and I want to introduce our next speaker. For those of you that have been around for the past hour or so, you already heard, but for those that are just joining now, our next speaker is Dr. Raul Andino. He's a professor in the Department of Microbiology and Immunology at the University of California, San Francisco. He's a virologist and professor, and he is working on some similar topics. And so we want to invite him to present his perspective on omics and virology, and specifically how the pandemic has also changed approaches in that field. So Dr. Andino, over to you.
Thank you, Leonid. Elia, let me see. Can you see my slides?
Yes, we can see it. Yeah.
Okay, so this first slide is one of these wonderful experiments of particle interference in quantum mechanics. And to me, is one of those examples. It's beautiful just to see the movie, but it brings two things here. One is the time resolution that we've been discussing from the very beginning, the importance of understanding that these things are, really dynamic. The other thing is that this has, is a very simple observation that enables to develop models that tell us a lot about the universe. And to me, it's a fascinating process that we can understand what happened in a supernova, why we have never been there and will never be able to survive there. But really what I wanted to discuss is, science and technology in the time of pandemics. And this is the type of technology people used in the 14th century when in Venice, the plague hit this, the bubonic plague. And really, right now, what's happening is that technology has changed. And what we're dealing with is, in the 21st century, a new type of pandemic or epidemics, but now we have additional tools, additional information that can help us to perhaps anticipate, predict, and intervene in a more effective way. And so really the concept that we trying to explore is how can we integrate the different layers that you will have when you are trying to understand virus diseases. And really, it goes from the epidemiological situation to the individual situation, to the tissue situation, and to the single cell situation. And of course, at the end of the day, it's all about molecules that somehow interact with each other and that determine all these levels of complexity. And the continuum between these molecules and the epidemiological effects, they actually break at some point. The question is, how can we integrate, how can we connect one layer with the other? It's a little bit the same with physics particles and large objects like the apple, that's famous apple that fills in Newton's head. How do we connect what happened in our universe with what happened at the particle level? And what we're trying to do in really scratching the surface here is both in cell culture and in animal models, we try to, on the one hand, understand the virus population dynamics. So we're trying to understand how the virus, by sequencing viruses, genomes, how the virus's populations differentiate in different organs. So here we have organ A, B, and C, which may be the brain and the liver and the kidney. And these populations acquire some sort of signatures that we can identify. And that's the experimental data that we can collect by sequencing viruses obtained from different organs or different conditions in cell culture. Then the important thing is statistical learning that allows us to classify what are the changes in the viral genome and how this viral genome is changing over time. So to give us some sort of dynamics, not only we have a classification of the alleles that we see in different organs, some will be beneficial, some will be detrimental in the organ A, but in B, it could be the opposite. Maybe the ones who were detrimental in A now are beneficial in B. So understanding this sort of, different changes and integrating that with the global gene expression. What happened to the ecological niche where the virus is replicating? What happened in organ A, B, and C is what will give us a little bit more understanding of what are the forces that are the virus is subject to change when it goes in A, B, or C. And one of the things that comes very clear is that if we think about ecology of infection, in other words, the infection as an ecological process, and really the process inside of the infected individual as an intra-host adaptation process, really as an evolutionary process, that ecology has give us a lot of guidance of how to analyze this kind of dynamic process. Really, there is a new organism getting into the niche, is modified by the niche itself because it's changing over time, but it's also modified by other individuals in that niche. And that's one of the things that I wanted to discuss is, how these defective interferon genomes can be playing a role in changing the outcome of the infection in individuals, infected individuals. Now, for all of you, you know by now that one of the important things in virus evolution and adaptation at the small level, meaning the virus in a test tube or in an organ, what happened is the virus acquire mutations because their enzymes that duplicate or replicate the genome, they are a little sloppy and they like to be sloppy and they introduce mutations, they make mistakes. And so you end up with a bunch of mutations in the genome. And really this is a population of genome that we call quasi-species. The other important mechanism of during replication is recombination, whereas one genome here in red is being read by an enzyme, which is a polymerase, and at some point the polymerase decides to leave this template in red and jump on a blue template. And so now at the end, as a consequence of that, you end up with a recombinant, a little bit of red and a little bit of blue. So understanding the interaction between mutations and recombination is critical to understand diversity in the virus population. And then there is selection. So evolution works as diversity and the product of diversity and selection. And of course, many of these genomes here, they are not viable. They have lethal mutations. And some of them get selected over time. Using sequencing, you can now start to classify mutations that you observe in these populations as a lethal, deleterious, neutral, if they don't change the fitness, or beneficial in a particular environment. And of course, these mutations are important to classify it. Not only, you cannot, again, it's the case of the dynamics of this process is very important. And the question is, this happened at a very micro evolution level. This happened at the level of the virus. The virus, when replicating in a particular cell, is introducing these mutations, recombining and being selected. But how that reflects what happened in a global pandemic is a big question. And can we use data obtained in very well-defined experimental conditions to understand and predict how a pandemic will evolve. How, is this virus going to evolve, or can you anticipate, whether it's going to be, what will be the next pandemic? That's a big question. And I think the answer really is, most people would say there is so much stochasticity in this process that the prediction will be very hard. But I think if one can reduce the problem to say, these are the things that cannot happen. We don't know exactly what's going to happen, but we know that the virus cannot go in this trajectory. And I think this is an interesting challenge for all of us. We developed, and I remember back in I think it was 2012 or something, that we sit together with Leonid in this coffee shop in the Weizmann Institute in Rehovot in Israel, and discuss about this technology. And Leonid, in I think in 24 hours, developed an algorithm that allows us to move forward much faster and more efficiently. Let me just walk you through what this idea is. One of the problems in biology is that we have experimental noise, and sequencing has an experimental noise, meaning that every time you sequence the DNA, the machine that is sequencing the DNA is introducing mutations. And at the end of the day, you don't know whether these mutations were in the viral genome or were introduced by mistakes during sequencing. So what we apply here is one technology that we call circular sequencing, or CIRSEC. What basically is we circularize the viral RNA, and these circles can be used as a template to create standard repeats when you do your cDNA synthesis, which is the first step that you need to do for sequencing. Then you put these cDNAs that are linked together because they've been produced in this circular template, and now you can do consensus, or really consensus sequence of all these repeats. And so mutations that are in all the repeats now are predicted to have been in the original template. This reduces the error of sequencing because you identify mutations that were generated there randomly, and it allows us to reduce the noise very, very much. Every time you read this is multiply the error of sequencing several times, so it's elevated to the number of times that you read these repeats. And this allows you to not only identify many mutations that are present at high frequency in the population, but many mutations like lethal mutations that are not present in the population can be also identified. And those lethal mutations are important because this allows you to say the viruses will never acquire this mutation. And maybe these are regions that cannot change. They are good invariance regions that you can target pharmacologically, for example. So what happened with the SARS-CoV-2? I know that many of us, all of us, I guess, have been very much affected by this pandemic, and we all, starting to get more familiar with terms like mutations and surface proteins and the spike and antibodies and so on. So I just wanted to, bring this things a little bit because, everybody's interested, and I wanted to use it as an example of how can you use the bioinformatics and omics to understand pandemic and maybe generate effective interventions. This is the situation globally and the US. You see these peaks that appear on number of cases, and this is consistent with the number of deaths. And a vaccine has been deployed since the beginning of 2021. And you can see that vaccines are effective, and yet there is again a spike. And those spikes emerge for a number of reasons. One is because not everybody gets vaccinated. Two, because the vaccines are not 100% effective, particularly are not effective at preventing transmission. And three, because the virus is changing. based on the challenge that the virus have in terms of transmission, replicating, and be successful at the replication, the virus is changing. And really, the critical thing in any of these viruses transmission is the ability of the virus to be transmitted from one individual to other. But as I said, the virus that you acquire is different than the virus that populate the different organs. Is probably different than the virus that really cause the disease. Because in many ways, those viruses need to adapt to a particular organ. For example, in the case of SARS coronaviruses, we know is a very complex disease that not only replicates in lungs, but also replicates and produce inflammatory effects in pancreas and in the heart. So we don't really understand many of this, and in part is because we don't understand this adaptive capacity of the virus. And the same thing with transmission. What happened with the new strains? This is an example, for example, in the UK, where you see that there was this strain here replicating until November 2020, and then there is another strain that replaced the previous one. So the question is, this strain, what is why is this strain replacing the previous one? Probably it's more fit, and that's a logical conclusion. But really the question is, why is more fit? And many people are focusing on this particular interaction. And in my mind, it's a little bit minimalistic and a little bit maybe unrealistic to just think that the only thing that can guide the virus to be more fit is the interaction between the receptor called AC2 and the spike. This is the little protein that is in the surface of the virus. And many of these mutations indeed map in this surface, and people have actually learned that some of these mutations increase or improve the interaction and recognition of the AC2. But really, when you think about it, there is another domain in S that is called the N-terminal domain, which is very different than the one interact with AC2. And there are many mutations here that emerge during the emergence of these new strains in the UK, in South Africa, Brazil, California. And now the Delta that also contain mutations in either of these domains. But importantly, we know that this protein, and this is the viral genome here, this protein is encoded here in S. But if you look at that, the viral genome is huge compared. So this guy has about 5 kbs, but the viral genome is about 30 kbs. And so focusing just in a small domain here in S might actually be a mistake. What else is going on? And we know now that from 28 viral proteins, 7 have been recognized to modulate the immune response. And that's very important because viruses, when it gets to the next ecological niche, the critical thing is that they were able to deal with the antiviral defense mechanisms. Not only they have to replicate, but also they need to block, suppress interferon or any other type of antiviral response. So, and there are mutations that occur through the genome. The question is, how do we integrate? How do we can understand these mutations? So I know that I'm talking more time than what I should. So let me ask, how much?
You have time. You have another few minutes. You're good.
How many minutes?
Another 10 minutes.
Okay. So let me go very quickly because I wanted to share with you 2 different things that we've been doing recently. And one is trying to use mathematical models that honestly, I learned from the interaction with Leonid and Simone Bianco, two people that I interact over many years. And what we wanted to do, and I wanted to put this is, very top of my inverted pyramid, the epidemiological aspect of this. And that is, how is non-pharmacological interventions can help to provide a solution to this epidemic. And the important thing is, can we define parameters to drive the epidemic? Can we train the model with real empirical data and then use additional data to try to see whether it fits? And how we can examine elements that could change the course of the epidemic in ways that are effective to provide these solutions. And so what we use here is very simple stochastic model, and basically it's like playing with a Pac-Man system in which individuals, they actually move within a restricted area in a surface, and every time they bounce against the wall, they change directions and they move that way. That's the simple, this is one individual. And then you can, of course, change the number of individuals, the size of the environment. And so what we are interested in is understanding the dynamics of the epidemic. And what we consider is that when two individuals, one infected here, circled by red, contact one individual that is uninfected at a distance that is lower than 2 radius, so they are really in contact. Now this guy has a probability to be infected, and over 10 days it recovers. So let me just show you the key here. Those are susceptible individuals. And what we're going to try to understand is the effect of mask in the probability of transmission and in the course of the epidemic. So in green are individuals that are susceptible with mask. And then the red circle indicates, individuals that are infected. And the probability of one infected individual to transmit the virus is given by this factor alpha that we calculate. It's basically a way to determine the probability of infection, and it has a specific number that has been obtained based on experimental data. Now, if one of the individuals, the infected individual, is using a mask, the probability of transmission is reduced by this number here, 0.05. And if the mask is used by the susceptible individual, the mask is used by the susceptible individual, but not by the infected, then there is another factor of 0.15 that determine or reduce the probability of infection. The important thing is that this type of exercise enables to have a visualization of the process. And we as primates work best at when you have a visualization of the process. That is what inspires us the most to understand a process. Numbers are more difficult sometimes to understand. So let me remind you, we have contact, infection, and recovery. And depending if you are using mask or not, you have a probability of being infected that is determined by this alpha factor. And so this, what you will see here is a panel where nobody is using mask, a panel where 40% of people are using mask, and a panel where 80% of people using mask. And in this kind of stochastic models, of course, you can change the probability of infection depending on many factors and adjusting the model that way. You can change the number of people using mask. And so what you see now is the people moving around, and there's individual people infected person and this person move around and see, as you see, this group of people with no mask get infected very quickly, and they have this blue peak here that is the number of people infected per day. And that's a critical parameter because the important thing is that you don't want to overwhelm the health system, the hospitals. And so the flatter this blue curve is, the better you can deal with the epidemics. And you can see that if 80% of people use masks, that blue curve is flatter than if nobody's using masks. Now, the other thing that people have been considering is this social distancing. And the way we model social distancing is, two individuals may be moving with no social distance, so they move around randomly, both of them. But when there is some social distancing, some proportion of the population does not move anymore. And that reduces the probability of encounter, the close encounter between the infected and non-infected individual. And so this simulates what would happen if you don't go to work, you stay home, or or you decide that you don't go to the grocery store so often, and so on. And when we use this, now you will see the same parameters: greens using mask, blue not using mask, different proportion of mask use here. And you will see now when I start the movie that part of the population does not move. In this particular movie, we're using 40% of people are staying home. And what you see now is, there's many people that are sitting in their homes, but there's other people that are bringing the pizzas or whatever they are actually coming to the thing. But of course, because there is less chance that they will encounter each other, you see that there is a reduction of the number of infected individuals per day. So the blue curve is a little bit smaller than the ones I showed you before. But the best effect is when you combine both things. Of course, use of mask reduce probability of infection, and the people staying put also reduce the probability of transmission. At the end of the day, this is a combination of these two studies. Here you have in red is wearing mask, and blue is no wearing mask. Blue is 40%, 80%, and here is social distance 0. This is very bad situation because you have a peak of individuals coming out at a particular day to the hospital, so you overwhelm the hospital. As you increase social distancing, you reduce that red peak. But if you use mask, which is 80% in green, basically, you have very little numbers of individuals infected. Now, really the question is, can you maintain 80% of use of mask and 80% of distancing? And the answer is obviously there is no compliance of using masks, and then social distance is being having an impact on society and economy and so on. But I just wanted to bring this at the end to these two graphics. These are deaths per million people, and you see that if you compare a curve where there's no social distance, or 80% of people is not going to work, the two curves seems to respond to the use mask. This is 0 to 80%. You can see that even though nobody's stay home, the mask really have a strong effect on reducing the number of deaths of people. But that's not the case if you invert the situation. Now you're looking social distance here and wearing mask 0 or 80%. Here is very high, but really the social distance, if you don't wear a mask, doesn't seem to be affected as much. And also, of course, the best is the combination. but the mask seems to be more effective. The more people use masks, more clearly you change the probability of a pandemic to expand. Well, I wanted to talk about the second part, which is the defective interferon particles, because this and I will try to do it in the last 4 or 5 minutes, just touching superficially, very high, the idea of how these defective interferon particles can be used as a therapy. And recombination is not a perfect process like any biological process. And sometimes the polymerase doesn't jump exactly to the place where, at the same place but in the blue genome, but sometimes it jumps forward. And Leonid was mentioning that often we see deletions in these viral genomes. And so we wanted to understand that, and we create a deletion in one, and this is a picorna virus, to create this defective interferon particle, or DIP. And the question was really, can we create a therapy using this thing? Because this defective interferon particle might actually attenuate infection. And this was a collaboration between Judith Friedman of Stanford, Leonid Brodsky from Haifa, Rob Nakamura from Aleph Biotech, and Simone Bianco from IBM, and Ilya Brodsky. And I think it's interesting because here there was 3 academic institutions interacting with 3 companies that provide different aspects. And it was a very interesting, not only a mixture of expertise, but also coming from different perspectives and areas. And basically, if you co-inject polioviruses with this defective interferon particle, you protect. Here is the co-infection, and here is if you just infect with the virus. You can see that you protect this animal. About 30% of the animals do not survive just with polio, but more than 80% survive. And that is because the virus cannot go to the brain. So here, the brain in solid lines, you see a lot of virus in the brain at day 1 and day 3, and this is why these animals die, because polio is a neurotropic virus. But if you co-infect with the ETIP, you see no virus in brain. And this also protects against other viruses, Coxsackie and influenza, and also protect against SARS-CoV-2. And here you are doing an intranasal inoculation of this tip, and then challenge with SARS-CoV-2. And these are highly susceptible animals. You see a reduction of about 3 logs of virus in the lung and the brain. And you can see that the virus replicates very well. This is the immunofluorescence using antibodies against the spike or N or spike, which are those 2 proteins of the virus. This is 3 days post-infection. This is 6 days post-infection. And when you do the co-infection, you completely protect both the lung as well as the brain. And this therapy works not only by prophylactic, putting the defective interferon particle before, but you can also use it therapeutically. So in this case, we are putting the ETIP 24 hours or 48 hours after infection, and also works in different strains that are circulating. This is the UK strain, this is the Californian strains, this is the initial US strain. Now, this is what I really wanted to tell you, because I think at this point, we wanted to understand, what was the nature of the protection, why this thing protects. And it turns out that we did a very initial analysis using RNA-seq data, and it's very clear in this volcano plot that there's a bunch of genes in red here that corresponds to genes of type 1 interferon response. And we use also animals that don't are not able to respond to interferon, and in that case, these animals are not protected. So the conclusion really is that these defective interferon particles do not work just simply by competing with the wild-type virus, but actually create some sort of antiviral state that help to eliminate the pathogenic virus. And basically, now we've been doing some analysis, and we need to go now with single cell analysis, but this is just flow cytometry. And what we can see in this analysis is that the virus is affecting the way that the immune system responds to the infection. There is some cells, specific cells, that now are present in the lung during infection that were not present or suppressed by SARS-CoV-2. So really, what at the end of the day, what I think is this type of therapies is doing is changing the character of the immune system from an inflammatory condition, which are represented by this type of cells. And we know that SARS, one of the reasons why SARS causes disease is because it initiates inflammatory response. We think the ET, this defective interferon genome, what it's doing is changing to an antiviral state. And then coagulation is another characteristic of the SARS or the COVID-19 disease. And we think that is the balance between these two lineages of immunocells that are very important to control. And age also, when you have, when age is another factor of risk. And what happens is when people get older, there's a problem at the stem cell level where you start getting more myeloid progenitors rather than lymphoids. And so again, understanding this process and this balance and this at the level of cell population will be very important for this particular disease. But there are many infectious diseases, particular virus diseases, in which the disease is the consequence of an imbalanced immunological response. And I think I'm going to have to stop here, although this last part is very exciting and it's something about we are developing with Ilya Brodsky, but I'm not going to have enough time, I believe. So, I'll stop here and I apologize for being a little bit longer than I was anticipating.
No problem. It's very exciting talks, and thank you for covering in such detail.